right, hello everyone, and I want to thank you for attending today's webinar entitled Achieving Deeper Insights into Complex Cellular Models Through Optical Enhancements to Automated Confocal Imaging. Our speaker today is Dr. Ernest Heimseth, an application development scientist here at Agilent Cell Analysis Division. Please click on the speaker module, speaker bio, in the top right corner of your screen to learn more about Ernest. Now, before we begin, I just want to cover a few housekeeping items. On your screen, you will see multiple application engagement tools that you can use. All of these tools are resizable and movable, so feel free to move them around to get the most out of your desktop space. Today's presentation will be played in the Media Player module, and it will be followed by a live Q&A session with Dr. Heimseth. If you have any questions during the webcast, please submit them when you think of them through the Q&A module located at the bottom left of your screen. We will do our best to get to as many of your questions as possible at the end of the presentation. The rest can be answered later via email. Please know that we do capture all questions. Additional help materials are available in the resources list located in the bottom middle of your screen. We encourage you to click on any links that you might find useful, including a relevant application note, technology pages, and more information about the BioTek Cytation C10 confocal imaging reader that you will learn about today. For the best viewing experience, we recommend using a wired internet connection and closing any programs in the background that could cause any issues, because webinars are bandwidth intensive, and closing any unnecessary browser tabs will help conserve that bandwidth. For the best audio quality, please make sure that your computer speakers or your headset is turned on and the volume is up so you can hear our presenter. Additionally, an on-demand version of today's webinar will be sent to you by email in the following days. Again, I just want to thank everyone for joining us today. And with that, I will turn things over to Ernest.
Thank you, Bear, for that introduction. We recently launched some exciting new features that enhance the confocal imaging capabilities of our Cytation C10 imager reader. And in this webinar, I'll be discussing how they can improve the data quality of imaging-based applications. To start, I'd like to provide just an outline of the topics I'll be discussing today. And the first is just a general overview of the Cytation C10. I'll then describe some of the fundamental concepts of confocal microscopy, as well as give an introduction to the new features that were launched with the Cytation C10, which are water immersion and deep sectioning disk. I'll then describe some of the optical challenges that these new features address, which are the numerical aperture and refractive index. I'll then provide some examples to demonstrate how these new features improve imaging capabilities of both water immersion as well as the deep sectioning disk. And then I'll end with a brief summary and some conclusions. So the Agilent BioTek Cytation C10 confocal imager reader is a versatile instrument capable of conventional multi-mode microplate reading using a quad monochronometer and PMT-based sensor. It also has three different modes of light microscopy imaging: a transmitted phase and transmitted light microscopy using both phase and bright field, LED-based wide field microscopy, as well as spinning disk confocal microscopy. In today's webinar, I'll be focusing on the enhancements we made to the confocal imaging capabilities of the Cytation C10 and discuss the relevant biological examples. So since the inception of confocal microscopy, a range of technologies have been used to achieve confocality within a microscopic image of a specimen. These techniques typically make use of a pinhole within the optical path. The Cytation C10 uses a rotating spinning disk with an array of pinholes, as shown moving around here. These spinning disk confocal systems provide distinct advantages over single-point scanning systems by allowing the user to decrease excitation energy to illuminate their sample and reduce phototoxicity and photobleaching. And this is important when imaging live samples where sample perturbation with illumination light can be phototoxic. The confocal optical system in this Cytation C10 includes spinning disks with either a 40 micron or 60 micron pinhole size, as kind of shown here. This is to enable confocality over a range of magnifications and samples. So the new features introduced in the Citation C10 address optical issues that really arise in two different areas of this entire optical path. One is in the interface between the objective and the sample, as well as optical issues involving the spinning disk and the pinholes itself. Now, one of the newest features of the Citation C10 is implementing automated water immersion imaging. This is accomplished using microfluidics, which deliver deionized water to an adapter cuff fitted on top of the water immersion objective, as shown here. This ensures that that bolus of water is maintained throughout your entire imaging session. We offer two different water immersion objectives, the 40x and 60x. Here I'm showing some of the specifications of each, and you can also find these online if you want to further understand their properties. Now, automated high-throughput water immersion imaging in the Citation C10 is possible with a continuous water column that is maintained throughout the process using that microfluidics setup. And this is capable in both 96 well and 384 well glass bottom or film bottom plates. Now, the other feature is the deep sectioning disc, and this is used to reduce background and thick biological samples, allowing a clearer view into that sample. This increased clarity enables you to collect more meaningful, detailed data from that sample. We'll go into this in a little bit later in the webinar. What I'd like to do next is just describe some of the optical challenges that the water immersion seeks to overcome, water immersion and the deep sectioning disc. Now, these two are essentially inefficient light collection, which is due to the numerical aperture, and well as Z's distortion of the image, which is due to the refractive index. And we'll just walk through and what those mean and how that affects your imaging. The numerical aperture describes light gathering capabilities of an objective. So if you consider this object right here, this spheroid on a sample, even though you're collecting an image from that sample, the emitted light is actually being emitted in all directions. And you're just catching just a fraction of that light that's being emitted, that's returning back to your objective. The degree of the amount of light that you're collecting is depending on the half aperture angle, or alpha, which is described here in this equation. So the numerical aperture is influenced by a combination of both the refractive index, which is n, multiplied times the sine of that half aperture angle, alpha. So we look at an example here with an objective of a numerical aperture of 0.6, where you collect a certain amount of light, and using imaging these 1 micron diameter tetraspec microspheres. When you compare the amount of the signal coming from that object to an objective with a higher numerical aperture of 0.95, you can see you're collecting more light. So higher numerical aperture objectives collect more light and are ideal for if you have a dimmer sample. This is very quantitative, so if you were to draw a line scan over that bead, what you can see here is that you're quantitatively collecting more photons, more light. Now, I mentioned there's two aspects of the numerical aperture. There's not only the half aperture angle alpha, but there's also the refractive index of n. So they kind of play into each other based on that equation. Now, the refractive index is a measure of how any particular material influences light trajectories as light is passing through that material. And so what water immersion seeks to do is to correct the light path of light as it traverses through all those different materials. As you can see on the left here with an air objective, is as light passes from your sample through, say, a cover slip or the bottom of a well, it's going to pass through at some degree. But then when it hits a material such as air, which has a different numerical, different refractive index, it will cause light to diffract and cause light to be less efficiently collected. Whereas water immersion, shown on the right, seeks to correct for that diffraction. And what this looks like is something like this. So these are those same 1 micron beads that are actually, this is actually a Z stack where you're going from the bottom of this image is the closest to a microplate well going into a depth of about 250 microns. Now, what you can see is a few different things. As light goes deeper into the sample of an air objective, you start to lose signal. As you can see that some of those beads at the bottom of the well are brighter than the ones deeper into the well into the Z direction. But the other thing too, which may be somewhat subtle, but I hope not too much, is that those objects which are spherical in nature become more distorted the deeper you go, although they are distorted throughout the entire Z compared to that same sample image in a water immersion system. So what refractive index does is correct for some of these Z distortions. Now let's look at some examples, some biological examples. First is some adherent cells and how water immersion can improve signal detection with gentler imaging. So here are 2 different images taken of the same sample with the same exposure settings. And what you'll see is that the water immersion collects more light with the same exposure settings. You can collect that same amount of light with an air objective. However, you're also going to be increasing your exposure settings, which can lead to either phototoxicity or photobleaching. And again, the main point here is that water immersion collects that light with using gentler imaging, with just more efficient imaging. Now here's another example using HE1080 spheroids of a thicker biological sample where water immersion collects more light at a gentler imaging settings than compared to air. The other thing relating to the refractive index is that water immersion improves Z-axis compression. And it does this by matching the refractive index of your aqueous sample. So this is an individual HC1080 cell, which is embedded in the collagen matrix to keep it in place. And so on the top row of images, we're just seeing is a traditional XY image. Now, if you were to rotate that to look at the Z plane and render a 3D image, what you're seeing below is a rotation of that 3D rendering. And what having a refractive index mismatch does is compress a 3-dimensional object, which I hope you can see on the left here with the arrow. And so what water does is gives a better representation, a better 3-dimensional representation of that object. So these are individual spheres, and if we scale up to a larger sample, such as these MCSF 10A mammospheres, what you can see is that not only is the compression addressed, but you increase Z resolution here. And so we're looking at the different nuclei within that mammosphere. So not only can you see this on individual samples, but again, with the design that we have for the Citation C10, we're able to implement high throughput automated water immersion imaging. Now, to illustrate how high throughput water immersion imaging can be useful, what I'm going to do is describe a nuclear translocation assay where we are monitoring TGF-beta signaling pathway, specifically with the downstream SMAD nuclear translocation. And in this pathway, when you stimulate cells with TGF-beta or BMP, what that does is is trigger a downstream phosphorylation event for either SMAD23 or SMAD159, depending on the growth factor. Now, these SMAD then form a tri-complex with SMAD4, which then translocates to the nucleus where they function as transcription factors leading to downstream biological activity. Now, the assay setup is fairly simple here. What we're doing is we're stimulating cells with a growth factor and then quantifying how much of that phosphorylated SMAD has translocated into the nucleus using a antibody-based approach. And this is a collaboration that was done with cell signaling technology using their very rigorously validated phospho antibody portfolio. Now, once you can monitor and quantify the amount of nuclear phosphorylated SMAD, what you could then do is start building dose response curves and derive EC50s. So the nuclear translocation can be done in both a 2D and 3D model system. And on the left, what we're showing here is A549 cells that are adherent cultures. And on the right, on the left panel is your control. And then after an hour of treatment with TGF-beta1, you see this increase in nuclear SMAD23. Now, you can see that there's split panel, and it's the outlines in one half of the panel is how you can do the analysis in Gen 5. So we've identified the nuclei within that image, and we can quantify the amount of SMAD23 signal within each nuclei here. As this is done in there, and then scale up to a larger model is A549 spheroids. And same idea is we can treat those spheroids with TGF-beta, fix immunostain, and quantify the amount of SMAD, phosphorylated SMAD that is translocated to the nucleus here as well. Now, in both of these cellular contexts, whether in adherent cultures or spheroids, one of the challenges with this particular assay in this particular system is that these are low abundance proteins, and so therefore the signal is very faint and requires a large amount of exposure. However, with a high throughput water immersion imaging setup, what we can do is not only image across a full plate at multiple concentrations, but we can reliably and very efficiently collect dim sample from those, all the way from the highest concentrations down to the lowest concentrations. And what we're able to do with that is start building those dose response curves and deriving EC50s from it. So on the left, what you see are those dose response curves from two different cell types done on the same plate using water immersion imaging. And on the right, what we see is A549 spheroids, where we can, again, do the same thing, is image multiple spheroids within a well or within a plate and derive very statistically robust data sets here. Now, moving on from the water immersion capabilities of the Cytation C10, what I'd like to do is transition over next to the second feature which we've introduced, and that is the deep sectioning disc. Now, to understand what the deep sectioning disc is capable of doing, I'd first like to explain a few aspects of the optical path. So the confocal pinhole, back to confocal imaging, refines emission light in the focal plane. So if you're imaging the spheroid, say, and you're on the focal plane is at the bottom of the spheroid, confocal imaging means you have placed, means you're able to place a pinhole in the conjugate focal plane. Now, for simplicity, I'm just showing a single point that is passing through a single pinhole, where in fact, this is happening across the entire disk. So if you rotate that, you'll see that the light coming from that point source is passing through that single pinhole. However, keep in mind that when your excitation light being laser LED based, you're still illuminating all the way through the thickness of that sample, which means that out of focus light, out of focus fluorophores are still being illuminated and so emitting light, which will appear as background because those fluorophores are not in the focal plane of your pinhole, and so that light then spreads out and bleeds through to neighboring pinholes called pinhole crosstalk. Now, what that looks like if we consider this HT10A spheroid, and this is a single plane about 50 microns into the spheroid, is that not only are you imaging the in focus plane here with the cells that you can see, but you're also capturing some of that out of focus light that's bleeding through from neighboring pinholes. So the solution to do that is to take that disk, that array of pinholes, and space them out a little further. That way you can refine that image and eliminate much of that out of focus light. So what the deep sectioning disk, given its name, really excels at is thick biological samples, such as spheroids, organoids, or tissues. And so here's one example. This is a mouse seminiferous tube. This is actually taken using a 20x objective. So we've stained the nuclei with Hoax 34580, as well as 2 different phalloidins and 2 different channels here. Now, this is a 100 slice maximum intensity projection. And so what you can see is using the regular space disk on the left, you get a high degree of background. That background is dramatically reduced with the deep sectioning disk. Another example here, and as this movie plays, this is playing through the Z slices of this tissue. This is a mouse stria vascularis. This is tissue found in the cochlea of your inner ear. This is a vascularized band of tissue that essentially maintains the ionic balance within the cochlea and allows to the hair cells in your inner ear to function correctly. So what you can see is as this pans through the bottom of this tissue through the deepest parts, the deep sectioning disc eliminates a lot of that background. And this, in turn, allows you to render a much clearer image of that. So this is the deep sectioning version of that tissue. Using the Gen 5 3D viewer, as you can see it rotating, you get a clearer view of all the vasculature of this kind of tissue. So to summarize, we've introduced some new features of the Citation C10. The water immersion with automated high throughput capabilities, the deep sectioning disk, which allows you to image thicker samples with much more clarity. These 2 features address 2 different optical issues, the numerical aperture and the refractive index. And I've shown examples of how water immersion can improve your imaging, as well as reduce Z distortion. And I've also provided some examples of how the deep sectioning disk can improve imaging of thicker biological samples. So in conclusion, the water immersion is capable of capturing more light with lower exposure times, and which reduces the potential for photobleaching. And this increased signal enables more detailed analyses without increasing gain or noise. The 60 micron deep sectioning disk diminishes crosstalk in thick samples, allowing a clear view into the sample. This increased clarity enables more meaningful, detailed data to be collected from the sample. So thank you very much for tuning in, and please stay connected with us. And at this time, I'll be happy to take any questions.
All right. Well, thank you, Ernest, for your terrific presentation. Now we'll address some questions that came in. If you haven't already, please submit them, your question now using the Q&A module. It's just a little box on the bottom left of your screen, and we'll get to as many as we have time for. So let's begin. We have, how thick of samples can I image with the water immersion objectives?
Yeah, so with the, can you hear me okay before I start talking?
Yes,
okay.
Yeah, I can. Yep,
okay,
thanks.
Good. It's a little tricky with the water immersion objectives because there's the working distance that you have for water immersions, which are more limited when you compare it to an air, but equivalent with immersion objectives such as like an oil immersion ones. For say the 60X, actually they're both about the same, or just around 250 microns. That's about as thick into a sample as you can image. So depending on whatever sample you have, be it a spheroid or a tissue, you can expect that kind of range of being able to image that deep into your sample. That being said, there's actually, because this is an automated system, there's a lot of checks in place to make sure that you avoid some of the dangers with a manual microscope setup, such as there are checks in place to prevent you from crashing your objective into your actual sample. So as you're toggling through, you physically can't go beyond the working distance of your objective to prevent those kind of issues from damaging your objective. An additional safety feature with the water immersion Cytation C10 is it's constantly checking to make sure that optical path is clean, in the sense of when you're dealing with an immersion system, you could introduce a bubble as you're flushing through the water. So if something's off with that, or if a bubble gets introduced, you'll be able to correct for that by priming or flushing more water to try to remove that bubble. So I kind of answered two questions there, but yes, a little over 200 microns based on the specifications of the objective itself is the working distance for that objective.
Perfect. All right, thanks. Let's see, a couple more came in. The Cytation C10 can house 2 disks, with the second being an option of either is that 40 nanometers or the deep sectioning disk. How do I choose which one of these to get?
Oh, yes. So that's an interesting question. That's a great question. Yeah, so you have an option. So now with the deep sectioning disk, you have an option to choose between those two setup. And a lot of it comes down to the fundamentals of confocality, is that the size of that pinhole, the diameter of that pinhole, really dictates how much of out of focus light you're able to remove. Now, the tricky thing about spinning disk setups is that you're kind of stuck with one diameter pinhole, either the 60 micron or the 40 micron pinhole. And the 40 micron one, it really excels if you're imaging a lot at using a 60X objective. The deep sectioning disc advantages are really when you're imaging a diverse type of a diverse amount of sample types. It's very versatile across magnifications too. So if you're going to be imaging things all the way from adherent cells up to larger, thicker samples, perhaps the deep sectioning just may be more versatile for you. If you have a very focused sample type that you need to develop, you can, but you want to eliminate as much out of focus light as possible with a confocal system, perhaps consider the 40 micron disc. Both come with the with having to optimize the acquisition settings, along with spacing out the pinholes or constricting the diameter of that pinhole. You are going to lose light, both at the excitation end, but also at the emission end that you're trying to detect. So you also have to consider that into play as well.
Gotcha. That makes sense. All right, a few more coming in. Can I still image with other objectives when the water immersion objective is in place?
Yeah, so nothing's really changed. Well, what hasn't changed with the C10 is that you still have the same 6-position turret with the Citation C10. Position 2 is dedicated only for the water immersion objectives, but all the other positions are available to put other magnification objectives in there. So you can still image with them. There might be some configuration challenges to have which position you can use, but you can still image with other objectives, even if you went alongside the water immersion objective.
All right. Let's see. Why did you say on a slide that 60 wells can be acquired per plate?
Yes. So with a microplate, you generally have like the skirts, or you have the actual underside of the wells, but the actual physical edge of the plates for many different plate types sits a little bit lower than the bottom of the actual well itself. And it becomes a spacing issue to be able to reach those outer wells of the plate. So the 60 wells that I refer to are the 60 inner ones. They exclude the outermost wells along the entire perimeter of the plate. So because of the clearance you need both for the water immersion cuff, but this would apply with any objective that's especially short working distance where you need to get really close to the bottom of that well. There becomes a physical barrier there. So as a safeguard, we generally try to exclude those outer wells, or say that there's limited imaging area with those outer wells.
I see. Perfect. All right, so let's see another one. Can kinetic runs be made with the water immersion?
Oh yeah, absolutely. So the example that I gave with a high throughput context or high throughput application was with fixed cells, fixed immunostains running through the entire plate. The same thing can certainly be done with the kinetic experiment. Again, it would involve some thinking through and planning out the experiment to figure out how fast of kinetics you need and how many samples or how many wells you need to image. But certainly, this is completely possible with water immersion.
Great. Let's see. We have another one here. Will this work for live cell imaging with different fluorescence dyes?
Yeah. So there are different challenging when doing live imaging experiments. Maybe because not only is the live, I guess the live imaging term is phototoxicity. So one thing that you would have to consider is how bright is the fluorophore that you're trying to image, be it if you have GFP or RFP or whatever have you. But also how long of an exposure, kind of back to that high throughput application that I described, is those are low abundance proteins, so they inherently needed a longer exposure. And those kinds of exposures are very challenging with live imaging, that you may introduce some level of phototoxicity. So you certainly can use this for live imaging. I think for any given experiment, you would certainly want to optimize with the brightness of whatever dye you're using, or fluorescent protein, as well as the robustness of the cell type or whatever live system you're using, to minimize those phototoxicity, photo bleaching conditions.
Would you recommend using the water immersion system or the deep sectioning disc for imaging organoids embedded in matrix?
Yeah, so it depends on it depends on your specific experimental setup and what you're trying to get out of it. So, like I mentioned in one of the first questions is the water immersion. The water immersion is available in 40 and 60x magnification for 40 and 60x objectives. So, one, you're limited to the image area. Fine, you can do a montage and build out a larger XY image, um, but you're still always going to be limited to that short working distance, which is around 250 microns deep. So if you want to be able to image a thicker sample that's beyond that, perhaps a deep sectioning disc is more appropriate for what you're looking at. Having organoids and cultured in a matrix poses other issues, optical issues, because you have this probably proteinaceous matrix that's also going to contribute towards light diffraction and other kind of things that would degrade the quality of an image in your system. Certainly a deep sectioning disc would help to counter that by eliminating a lot of that diffraction, a lot of that out of focus light. Again, back that pinhole crosstalk that I described. The deep sectioning disc can certainly do that. And you're not limited to water immersion by using the deep sectioning disc. The deep sectioning disc, you can combine that with a lower magnification for a larger sample. Like you could combine it with the 20x objective to just get a larger image, which also gives you a larger working distance as well.
Gotcha. Perfect. All right. Let's see. Is there image analysis software associated with the C10?
Yeah, so the focus here was certainly on the new features, which are hardware improvements, the water immersion and the objective and the deep sectioning disc. But on the software side, our Gen 5 software has an entire suite of analytical tools that you can use in combination. That was a small example of the high throughput nuclear translocation assay that I showed, which showed one of those where you can identify nuclei within the Gen 5 software and then measure in a separate channel fluorescence intensity or different parameters. But certainly there's an entire analysis package built into the software to help you not only take these images, but extract meaningful data from them, meaningful quantitative data from them.
Great. Thank you. Let's see. Looks like we got a couple more here. Have you tried the deep sectioning disc with plant tissue, for example, leaf tissue? And if so, how did it work?
I have not. I would have to cross-reference with some of my colleagues that may have, but I'm sorry, I can't really provide much insight here. Most of what I've worked with during this development is with more mammalian cells, or in the context of either tissue or cultured cells. But certainly, let's stay in touch, and we'll reach out to you individually to kind of circle back and provide some kind of insight for you on that kind, if that's the kind of sample that you're most interested with.
Perfect. All right, we have another. Which type of plates, either glass, plastic, or other bottom plates, should we use with this device? Any preference?
Oh yeah, so these samples, everything I showed you here was using the Agilent plate that we offer. It's a 96 well, it's a cycloolefin filmed material that's on the bottom. We found that it's very conducive with high resolution imaging, such as what we use it for the confocal imaging. The thickness is around that of glass, is about 170 microns, or like a 1.5 cover slip. You certainly could use glass if that's what you're used to, if that's what you prefer. With the water immersion objectives, we validated it with a number of plate formats and sample formats, cover slips and slides and things like that. And so what you'll see is you'll have a list to select from of those formats, of those plate formats, or the whatever sample formats that you can choose from that are specifically designed, or in the Gen 5 software, specifically designated for short working water immersion imaging.
Perfect. Well, that is all the time we have for today. I just want to, on behalf of Agilent Technologies, I want to thank everyone that joined us. If we did not get to your question, as Ernest said before, we will follow up with you via email. And if you would like more information on Agilent BioTek's products and services, including an archive of previous webinars, please visit us online at www.agilent.com. We hope that you enjoyed today's presentation and have a great rest of your day.
Thank you for tuning in.